Assalamu alaikum. It's my third lecture, but before starting the new topic, I would like to revise the previous one. In the previous lecture, we were discussing the humoral immunity that is carried out through the B lymphocytes. In that, we have the complement system, which is the combination of many other pathways. I told you that classical pathway, lectin pathway, alternate pathway, all these they are the components of the complement pathway or complement system. In this figure, I just want to show you that how all these systems they are interconnected with each other. This time I am showing you this figure because at this stage you have a good knowledge about the complement system. So through this figure you can appreciate their interconnection, how they are connected with each other. Classical pathway and lectin pathway combine at this stage while the alternate pathway at this stage. So that here in the end they are forming the membrane attack complex through which the lysis of the cell take place. One more thing you should appreciate here, that it is acting through C1q, it is through MBL and factor B and D, because in the next following slide I will show you these factors, this type of the proteins separately also. So at this stage I am highlighting these also, so that in the next you will easily recognize these. This is the whole complement system, and you can see they are not only connected with each other, but they are running other systems also, other processes also. As I have told you repeatedly that this is not so simple system, it is a very complex system. The same thing you can see clearly from this figure also, and this one very nice figure to show the interconnectivity of all these processes. And in the end I am showing you the lactic pathway also, after which the membrane attack complex is formed through which the lysis of the cell take place. These are the factors which I want to show you separately also for the classical pathway C1q, lectin NBL and foilin ficolin 3, and alternate pathway for the factor B and D. You can see the function of C1q also here in the animated form. Now, with a thorough knowledge of humoral immunity, I am coming to the cell-mediated immunity, which is carried out through the lymphocytes, T lymphocytes. Mainly, they are working as the B lymphocytes, and the main target is to kill the infective agent. There is a little difference between the function of the B lymphocyte or the mechanism of the B lymphocyte and the T lymphocyte. One important difference is that for the action of the T lymphocytes, we need some more assistance through specific protein particles. They are also working as the B lymphocyte. As they become activated, T cells also become activated. As the B lymphocytes have memory cells, same for the T lymphocyte, they have also memory cells. But the difference lies that T lymphocyte recognizes peptide antigen on macrophage in association with major histocompatibility complex. This is the thing which must be there on the antigen presenting cell, after which the attachment of T cell to the antigen presenting cell will be possible. So this is the difference, that for the attachment of T cell to the antigen presenting cell, this complex must be there, that is major histocompatibility complex. Three types of antigen presenting cells may be there. They may be macrophages, B lymphocyte or dendritic cells. Through this same figure, I want to show the function of the T lymphocytes. But you must recognize this difference regarding the T lymphocyte action, that now the antigen presenting cell, that is, this is the cell to which the antigen will be attached, but it has also MHC. It must be there. Only then this antigen and presenting cell will be able to be attached with the T lymphocytes. Once this attachment takes place, these T lymphocytes, they become activated and then comes to the circulation, and through the blood and the lymph they reach to the target organs. As we have in the B cells, memory cells there, same for the T cells, we have some of the basic memory cells there which come to the lymphoid tissues, and because of that we can have again the primary and secondary responses. As far as the MHC is concerned, regarding that we have again two types of MHC cells, type 1 and type 2. Type 1 present antigen to cytotoxic T cells, and type 2 present antigen to the T helper cells. You may be confused with these words, that are what are the cytotoxic T cells, what are the T helper cells. I will explain these later on so that you come to know what are these. At this stage we are discussing that for the attachment of antigen presenting cell to be attached with the T cell, we must need MHC. How it combines, how it is formed, major histocompatibility complex. You can see different types of the proteins, different complement system proteins. They combine to form C5b, C6, C7. Firstly, they combine, then C8 comes in contact with that, and finally the C9 that combines. All these combines to form the major histocompatibility complex. So what is that one? It is actually all these proteins, proteins combined together and form the major histocompatibility complex, which is of two type, type 1 and type 2. Through this animation, you can clearly see how it is going to be formed. This factor, that is C9, come in contact with other factors, and then many of these they combine to form the MHC. Now through this picture I want to show what is the next step. Once it is ready, that is the antigen presenting cell, which may be macrophage, B lymphocyte or the dendritic cell, either of these. Once it is ready with MHC, now the T cells, they are going to be attached with them through the receptor. This is the very specific cytotoxic T cell. So we here have the MHC1, and for the helper T cells we have the MHC class 2. So now I am telling you what are these cytotoxic cells and helper cells and other cells. Actually, the T lymphocytes, they are of three types basically. We can have helper T cells, cytotoxic T cells and suppressor T cells. Mainly the helper T cells, as their name shows, they are going to help this process, and they are going to help the other type of the T lymphocytes. Once they are activated, they cause the release of different types of the lymphokines. Interleukin 2, 3, 4, 5 and 6, CSF, GM and interferon, all these things, they are released through the helper T cells once they are activated. And as they are helper T cells, they are going to help the other types, that are the cytotoxic and the suppressor T cells. So most important thing is that they are helping or assisting the overall function of the immunity. So it is the helper T cell which are going to assist the whole of the immunity process. So imagine if they are destroyed, what happens? The whole system of immunity, that will be collapsed. And very specific disease which is called HIV infection causing AIDS. The other function of the helper T cells, the details are growth or proliferation of the cyto T cell and suppressor T cell, B cell growth also, macrophage activation also, stimulation of the T cell, positive feedback by interleukin 2. Now through this figure you can see how the helper T cells, they are going to assist the other types. They are releasing the different kinds of the lymphokines, which are not only the affecting the B lymphocyte but also have a great influence over the cytotoxic and suppressor T cells. Once they are acting on the B cell, they are going to produce the different types of the immunoglobulins. As I have told you that helper T cells are going to control or affecting the whole of the immunity process, and through the HIV infection they are destroyed. This type of the infection causing the very important disease condition which is called the AIDS or acquired immunodeficiency syndrome, as its name shows that the immune system is deficient. And imagine if your body immunity fails, your body is susceptible for repeated infection. If there is any slight injury or slight infection that cannot be treated by body immune system. So repeated infection is there, followed by fever and other complication, and may lead to the death of the patient because of the collapse of the immune system. So people with AIDS are highly susceptible to the repeated infection and cancer that take advantage of an immune system in collapse, because AIDS arises from the loss of helper T cells. Coming to the next type of the cytotoxic T cells or the killer cells. As its name shows, cytotoxic, it is very much toxic to the cell, so it is called as killer cell. These type of the cells bind tightly by receptor proteins to the foreign organism, infected cells of the body. They secrete a special type of the secretions, perforins, and make holes in cell membrane of the bacteria so that different killing enzyme, different specific enzyme, they flow into that and cause the rupture of the cell. Then detached from the previous one and ready to attack the next one cell, infected cell. So they destroy the cancer cells, virus infected, transplanted tissues and other foreign cells. Through this picture, I want to show you the function of the cytotoxic T cells, how they become activated and how they cause the lysis of the infected cell. Lastly, we have the suppressor T cells, and the other name is regulatory T cell. They serve the purpose of preventing the cytotoxic cell from causing excessive immune reaction that might be damaging to the body's own tissues. So what is their function? As their name shows, they regulate or suppress the other cells, mainly the cytotoxic cell. If there are no suppressor T cell, what happens? The reaction or the steps of killing bacteria by the cytotoxic cell that prolongs, so they may damage the healthy own body's tissue. To prevent that one, we need the assistance of suppressor T cells. So they limit the ability of immune system to attack a person's own body tissues, and that property is called immune tolerance. So what is that one? Immune system recognize body's own cells, and it is very necessary. If this not happens, once these cytotoxic cells or this process of immunity body defense starts, what happens? They may engulf the healthy own body tissues. So there must be a thorough check on these reactions to limit these reactions, so that they must engulf only the infective tissues. They must kill only the infective agents, leaving the healthy tissues. So this is our body own tolerance, and how it mainly occurs during the process of pre-processing of the lymphocyte in thymus. As I have already told you, in the thymus, once these type of the lymphocyte they are generated or the form, they go to the thymus to be trained how to recognize the body own tissues. they must differentiate between the healthy and unhealthy tissues. So they should be trained properly to engulf only the unhealthy infective tissue, leaving the body owns healthy tissues. So with the repeated lectures to these lymphocytes, they are trained. Once they come out to the thymus, from the thymus, they recognize easily what is own body tissue and what is infected foreign tissue infected through these specific agents. And one more thing, dual signals normally activate T cell. Body cells never have these dual signals. So T cells are not activated. This is again a very important point, that for the activation of T cells, dual signals there must be present. Normal healthy tissues, they are not sending dual signals. So normally these specific T cells, T lymphocytes, they are not activated. So in this way, our own body healthy tissues, they are prevented by engulfing by the action of these lymphocytes. B lymphocyte and T lymphocyte, they are prevented, they are reserved, and only the infective tissues, they are killed. So with this we complete the process of cell mediated immunity. In the next lecture, I will discuss the topic of immunity again, but here we have completed the humoral and cellular immunity to some extent. Next, I will tell you what is the active and passive immunization. Till that, Allah Hafiz, thank you, and Allah Hafiz.